Hello, good morning everyone. Welcome to UDAAN Career Institute. In today's class, we will continue with chapter 2, that is reproduction in angiospermic plants. Now, as all of you know that in the last class we have gone with the structure of pollen grains and viability of pollens. In today's class, we will start with the another topic, that is female reproductive organ. It is also known as gynosium. Now, as all of you know that gynosium, that is the carpel, it is considered as the female reproductive part of the flower. This gynosium, it consists of either single pistil or it can have more than one pistil. So, as all of you know that carpel, it is composed of pistil, and this gynosium, it may have one pistil. So if gynosium is having only one pistil, then such kind of flower, it is basically known as monocarpellary. Or if it is having more than one pistil, then it is also known as multicarpellary. So there are two types of flower depending upon how many number of pistils are present. Either it is single or it is more than one. If it is a single pistil, it is known as monocarpellary, and if it is more than one pistil, it is known as multicarpellary. Now if a flower consists of more than one pistil, then in case of multicarpellary flower, further there are two divisions. So in multicarpellary flower which have more than one pistil, if the pistils are fused together with each other, then it is known as syncarpous, and if the pistils are free from each other, then it is known as apocarpous. So multicarpellary flower is further divided into two types, that is syncarpous and apocarpous. Syncarpous flowers are those flowers in which the pistils are fused together, whereas apocarpous are those flowers in which the pistils are free. So if you see the diagram which has been shown here, the diagram B, it is a multicarpellary syncarpous pistil, that is fused pistils of papaver, whereas if you see the diagram C, then it shows a multicarpellary flower and it is an apocarpous gynosium of Michelia, where the pistils are free. So in case of B, diagram B, the pistils are fused together, that's why it is syncarpous. Example is papaver. And in case of diagram C, the pistils are free, they are not fused with each other. You can see all the carpels are different from each other, they are well segregated from each other. That's why it is known as apocarpous gynosium of Michelia. So that's how a multicarpellary flower is further divided into syncarpous and apocarpous. Now each pistil, as you know, that it has three parts: stigma, style and ovary. So if you see the diagram of a basically pistil, then the uppermost part is known as stigma, then there is a long stalk like structure which is known as style, and at the bottom there is a bulged part which can be seen here, which is known as ovary. So basically each pistil has three parts: stigma, style and ovary, where stigma, it is the landing platform for the pollen grains. The pollen grains, they are shed from the anthers and they land on the surface of stigma for the fertilization. So it is considered as the landing platform. Style is a slender part beneath the stigma. It is a long slender structure, so it is present beneath the stigma. Below the stigma, style is present, and at the base there is a bulged part which is known as ovary. Inside the ovary there is presence of ovarian cavity, which is also known as locule. So for us, ovary is very important. So inside the ovary, ovarian cavity is present, which is known as locule. Placenta is located inside this ovarian cavity. So placenta is there inside this ovarian cavity, and this placenta, it will give rise to ovule, which is also known as megasporangia. So placenta is located inside the ovarian cavity, giving rise to ovule which is known as megasporangia. The number of ovules in an ovary, it may be one or it may be many. So if it is one, the examples are wheat, paddy and mango, whereas if they are many, the examples are papaya, watermelon and orchids. Now if you see the diagram which has been shown below, the diagram A, it is the pistil of a flower hibiscus, whereas the diagram B, the diagram B, it is the multicarpillary syncarpous pistil of papaver. The diagram C, it is multicarpillary apocarpous pistil of michelia, and whereas the diagram D, it is an example of anatropous ovule. So it is a diagrammatic view of anatropous ovule, where you can see that there is presence of hilum, funicle, micropyle, micropylar pole, outer integument, inner integument. Inside there is presence of nucellus. Then in the center there is presence of embryo sac, and the bottom part there is the chalazal pole. So with this we will go with the structure of ovule or megasporangia. The fourth diagram which has been shown here, that is the diagrammatic view of an anatropous ovule. So here we will study the structure of this anatropous ovule or the megasporangia. Now as all of you know that ovule is present inside the ovary, so it is a very small structure. It is attached to the placenta by a stalk-like structure which is known as funicle. So the placenta, it is attached to the ovule by the funicle. So body of ovule, it fuses with the funicle in a region which is known as hilum. So hilum is basically a junction between the ovule and the funicle. So basically ovule, basically it is a small structure. It is attached to the placenta by a stalk-like structure which is known as funicle. So through this placenta, the nutrition it is given to the ovule. This ovule, it fuses with the funicle, and the fusion point is known as, or fusion region is known as hilum. So hilum is basically a junction between the funicle and the ovule. Now each ovule has either one or two protective cover or envelopes which are known as integuments. So either the ovule would be covered with one integument or the ovule would be covered by two integuments. Now in case of anatropous ovule, it has two integuments, and that's why anatropous ovule, it is a bitegmic structure. Because it has two integuments, it is also known as bitegmic. The integuments, it covers the ovule except the tip portion where there is a small opening which is known as micropyle. So if you see the structure of anatropous ovule, at the tip you can see that the integuments are absent, and as a result there is a small opening which has been formed, which is there is a small opening which has been formed here. You can see in the structure there is a very small opening which is present, and this opening is known as micropyle. The area where micropyle is present is known as micropyle pole, or it is known as micropylar end, and opposite to the micropylar end there is chalaza. So that part is known as chalazal end. So in anatropous ovule, the micropylar end and the chalazal end will be opposite to each other. So basically the chalazal end, it represents the basal part of the ovule. Now enclosed within the integuments, so inside the integuments a mass of cell is present which is known as nucellus. So this nucellus, basically it provides nourishment. So inside the integument there is a mass of cell known as nucellus, and this nucellus has abundant food material, and that's why it helps in providing nourishment. Located in the nucellus there is an embryo sac, also known as female gametophyte. So within this nucellus, the female gametophyte or the embryo sac is present. An ovule generally has single embryo sac which is formed from a megaspore. Now with this we will be starting with the another topic, that is megasporogenesis. So as we have gone in the last slide, that ovule has one embryo sac and it is formed from megaspore. So what is basically megasporogenesis? Megasporogenesis is the process of formation of megaspore from megaspore mother cell is known as megasporogenesis. So it is almost similar in case of microsporogenesis, where in case of microsporogenesis we can see that microspore mother cell was giving rise to microspores. Here in case of megasporogenesis, the megaspore mother cell, it give rise to it is obtained from megaspore mother cell, and this is known as megasporogenesis. So process of formation of megaspores from megaspore mother cell is known as megasporogenesis. Now inside the ovule, a single cell which is present at the micropylar region of the nucellus, it will differentiate and it will get converted into megaspore mother cell. So when you see inside the ovule, there will be a single cell which is towards the micropylar region, means it will be near to the micropyle. Now that particular cell which is near to the micropyle region of the nucellus, it will get differentiated and it will get converted into megaspore mother cell. This megaspore mother cell, it is very large and it contains dense cytoplasm and a prominent nucleus. This megaspore mother cell will undergo meiosis and it will lead to the formation of 4 megaspores. So this is how megaspore mother cells, they give rise to megaspore by the process of meiosis. So I'll repeat this once again. What is megasporogenesis? The process of formation of megaspores from megaspore mother cell is known as megasporogenesis. How this megasporogenesis takes place? If you see inside the ovule, there is an area which is known as nucellus. So one cell of the nucellus which is present near to the micropyle, it will get differentiated and it will get converted into megaspore mother cell, and this megaspore mother cell will undergo meiosis and it will give rise to 4 megaspores. So basically this process is known as megasporogenesis. In next class we will study megasporogenesis and development of female gametophyte in more detail. Thank you very much.